This procedure isolates specifically glycosylated glycopeptides from human plasma. Plasma is digested with trypsin to yield a mixture of peptides and glycopeptides. Glycopeptide standards, positive and negative controls for lectin chromatography, are spiked into the plasma. The sample is subjected to high-pressure, liquid chromatography-based lectin affinity chromatography. The lectins used bind to fucose and sialic acid, respectively. Peptides and irrelevant glycopeptides elute from the column in the flow-through fraction. Acetic acid is used to elute the bound fraction from the column. This is enriched for specifically modified glycopeptides.
Hi, I'm Penny Drake from the laboratory of Susan Fisher in the Department of Obstetrics, Gynecology, and Reproductive Sciences and the Sandler Moore Mass Spectrometry Core Facility at the University of California, San Francisco.
And I'm Eric Johansen, also from the Sandler Moore Mass Spectrometry Core Facility and the Fisher Lab.
Today, we will show you a procedure for performing lectin chromatography to enrich glycopeptides from human plasma and other complex mixtures.
We use this procedure in our laboratory as a general tool that has numerous applications. For instance, it plays an important role in our breast cancer biomarker discovery workflow.
So let's get started.
To begin this procedure, wear a mask to protect against inhalation of the beads, then weigh out 100 milligrams of POROS beads and transfer to a clean Eppendorf tube. Approximately 300 microliters of beads.